Today, we are going to discuss venous system of embryo. But before starting this, let's revise the basics of heart development together. This is embryonic disc with dorsal view. This is cranial end. And this is caudal end. The primitive streak first appears in the epiblast at the caudal end of the embryonic disc, extends towards its center, and then develops the primitive groove in its dorsal surface. At the cranial end of this develops the primitive node. Now, if we cut and look into it, if we cut it this way and look into it. Now moving forward, these are epiblast cells. And these are hypoblast cells. Now, epiblast cells migrate across the disc, through the primitive groove, and into the space above the hypoblast. These become the embryonic mesoderm, creating the 3 germ layers. Once the cells have invaginated, some displace the hypoblast, creating the embryonic endoderm, and others come to lie between the epiblast and newly created endoderm to form mesoderm. Cells remaining in the epiblast then form ectoderm. Now, this is a cross-sectional view. As we have said that this is ectoderm, this is mesoderm, and this is endoderm. So, during the 3rd week of development, in the lateral plate mesoderm has some changes. It forms other layer called as angioblast. That will form the heart tube. That will be explained later. And inner area called as hemocytoblast. Then there is the embryonic folding.
Sir, name indicates that it must be the folding of the embryo. But I am still confused. How is it possible? Does it cause malformation, disruption, or deformation of formed structure up till now?
No, Alize. It's normal development process of the embryo. Moving forward. So in the next step, there is formation of endocardial tube or heart tube. And this is pericardial cavity. Now, let's have a more close view of heart tube. This heart tube is made up of 3 layers. Outer layer is endocardium. That develops from that angioblast.
Oh sir, the lateral plate of mesoderm stimulates into angioblast and hemocytoblast. This angioblast then convert to endocardium.
Yes, Diana. You are right. Now, other layer is myocardium. These myocardium secretes a substance that forms a layer called as cardiac jelly. So, this was the cross-sectional view. Now if we move the heart tube out. I repeat. Now if we move the heart tube out. Can you imagine, class, how it will look like? So, let's see how it will look after moving it out of embryonic disc. So, it will look like this. This is the early heart tube at 22 day. This is truncus arteriosus. This is bulbus cordis. This is primitive ventricle. This is primitive atrium. This is sinus venosus. Bulbus cordis will move in this direction, while primitive ventricle will move in this direction. The heart tube begins to bend at 23 days and stops at 28 days. Now, this is heart tube at day 29. You can see the junction between the bulbus cordis and ventricle by the presence of the bulb ventricular sulcus. Now, this is truncus arteriosus. This is bulbus cordis. This is ventricle. This is left atrium. Now, we will talk about the sinus venosus. As we have discussed, this is sinus venosus. The sinus venosus comprises the inflow to the primitive heart tube. It means sinus venosus comprises the inflow to heart at early stage. So, this area comprises the outflow. And this side is arterial end. And this side is venous end. Now, what about the structure of sinus venosus? Well, medical class, let's discuss the structure and fate of sinus venosus. Sinus venosus has left sinus horn and right sinus horn. As you can see, each horn has further 3 extensions. So these horns are formed by the major embryonic veins, such as right sinus horn has cardinal vein, umbilical vein, and vitelline vein. Similarly, left side sinus horn has cardinal vein, umbilical vein, and vitelline vein.
But, sir, how to remember these 3 veins?
You can remember it using a mnemonic. CUV, C for cardinal vein, U for umbilical vein, and V for vitelline vein. Now, venous drainage becomes prioritized to the right side of the embryo. Well, the left sinus horn becomes smaller and less significant. This is cardinal vein, and this is vitelline vein. Now, as we have discussed that, the left sinus horn becomes smaller and less significant. The next step is that left sinus horn eventually forming the coronary sinus and draining the coronary veins into the right atrium. The right sinus horn persists, enlarges. Cardinal vein becomes superior vena cava, and right vitelline vein becomes inferior vena cava. Now, let's understand the venous system in detail. In the 5th week of intrauterine life, the veins of the body develop from 3 sets of primitive veins: vitelline veins, umbilical veins, and cardinal veins, which drain into sinus venosus.
Sir, we will revise them. First are the vitelline veins, which return poorly oxygenated blood from yolk sac. Second are the umbilical veins, which carry well-oxygenated blood from placenta.
And third are the common cardinal veins, which return poorly oxygenated blood from the body of the embryo.
Very good, class. Now, let's have a view of veins at 28 days. This is heart. This is sinus venosus. This is left anterior cardinal vein. This is right anterior cardinal vein. This is right posterior cardinal vein. This is left posterior cardinal vein. Common cardinal vein on each side is formed by union of anterior and posterior cardinal veins. So, common cardinal vein. This is right vitelline vein. This is left vitelline vein. This is right umbilical vein. This is left umbilical vein. So, these are veins at 28 days. The content we have covered so far concludes our current discussion. However, I am excited to inform you that our next video will be dedicated to a thorough and in-depth examination of the intricate topic of vitelline and umbilical veins. In this upcoming video, we will delve into the details, intricacies, and significance of both the vitelline and umbilical veins, providing a comprehensive understanding of their functions, development, and relevance. This upcoming video promises to be an informative and insightful session, shedding light on these crucial aspects of embryonic and fetal development. So, stay tuned and be prepared to expand your knowledge as we explore vitelline and umbilical veins in meticulous detail.
Upgrade your medical education with sqadia.com, the ultimate online video platform tailored for medical students and professionals.